recording going. All right. So we'll see how long my voice lasts. Something about going to a cross-country meet for little kids yesterday has given me a sore throat. I don't know if I just inhaled a bunch of grass clippings or if I have strep. Who knows? Nobody really knows at this point. Anything is possible, so we'll try to see how we do. So this should hopefully be our day that we finish up on resistance training. What we have left to do with resistance training is to dig in a little bit deeper into what we typically call the cellular mechanisms or the cellular signals and triggers that turn on protein synthesis in a muscle fiber, okay, that tell the nucleus to make actin, to make myosin, to integrate those things in with our existing muscle fibers and to make them bigger. Okay, so we left off on Friday talking about the idea of anabolism and catabolism, that in general, when we make more protein, that's called anabolism. And that catabolism is an instance where we're breaking down those proteins. Okay, so if we know then that resistance training specifically, okay, is going to be a thing that leads to an increase in muscle size, then resistance training must trigger something that drives anabolism, okay? And I guess conversely, if you think of things like as two sides of a scale, right, it could also limit catabolism, but it actually raises catabolism. Right, after about a resistance training, catabolism also tends to go up. You just need anabolism to go up even more. Okay, so we're going to get into kind of the cellular regulators of these things. Okay, so this next slide, we talked about the idea of what's called fractional protein synthesis. I think it was on Friday, but these are some data from sort of the early kind of mid 2000s, okay? And as I had mentioned that we talked about this. This is from the medical center at UT Galveston, and what you can see is this is what's called myofibrillar protein fractional synthesis rate, okay? And all this is it's a measure of what is happening inside the muscle cells, or what we think we can approximate from inside the muscle cells in a muscle after a bout of resistance training. So here's our resting fractional synthesis rate. The numbers, right, are a percentage of something per hour. Like, don't worry, the numbers themselves, the magnitude doesn't mean anything. It's the relative change that we see here that's going to be important. Okay? But here's some resting rate that like all of you guys might have now. We do about a resistance training. We measure 6 hours later, right? And it's gone up almost 3 times. We measure 24 hours later, and it's definitely gone up, a little bit more. And then it's going to begin about 88 and 72 hours to begin to come back down. So we see this scenario where a single bout of resistance training can increase in a particular muscle the protein synthesis rates for several days. Okay, for several days. That's the big thing here. All right. By comparison, and I don't have data on here, if you just eat a meal that has a reasonable amount of protein in it, you will get an increase in fractional synthesis rates that lasts about 24 hours. Okay, it lasts about 24. And so then there's been lots of studies where we're at rest, we mess around with what is your diet beforehand, you do your resistance training, and then we feed you different things after, right? We feed you things right before, during, right after. We mess with what's the concentration of protein, what's the concentration of carbohydrates, what's the concentration of fat, that kind of all work together to see if we can figure out what do we need to do to maximize this and get the largest possible response. And what I can tell you guys, if you really care, is the best data that we have so far is if you eat a little bit of protein in combination with some carbohydrates right before you work out, during your workout, and then within about an hour to maybe 90 minutes or 2 hours window afterwards, then we get slightly measurable, but slightly larger increases here. Okay, as you all will see next week when we talk about hormones, you need to combine the protein with carbohydrate because that stimulates insulin. And the insulin can function to be partially anabolic in that way. And the closest thing in nature, or nature's the wrong word. It's not in nature. Chocolate milk is not in nature, but I wish that it was, right? Ellis probably thinks that, right, you get chocolate milk from brown cows, and all of that. But chocolate milk has basically like the exact magic ratio of carbohydrates to protein in it that matches sort of what works best. So lots of supplement things that you can buy. Gatorade, I don't know if they still make them. They used to have different kind of variations of stuff. Muscle milk, some of the things like that. It's basically looks the ratio of carbs to protein looks like chocolate milk in that way. So there is something to all of that. We can make this a little bit bigger, but right, like this goes up like threefold, fourfold. We're not going to get a 3 or 4-fold increase in the size of your muscles. So remember, this is the single bout sort of response. Okay. All right. So the rate goes up, and there has been some question, right? We're going to get this rate. Are we going to make these new proteins and incorporate them into the existing muscle fibers, right? Or are you going to make new fibers? The ones that you have are going to be there, but you're going to make some new ones. What's going to happen to allow our muscles to get bigger? Okay. And so that's what this slide is going to try to illustrate for you. It's this idea of something called hyperplasia. Okay. And so hyperplasia is the idea that I have one muscle fiber and I train it, and then poof, it splits into two fibers. Okay, that's hyperplasia. We don't think in humans that the data support this as the way that our muscles get bigger. If you look in some animal models in cats and mice and rats and things, if you load their muscles in certain ways, especially if you do an overstretch model of inducing hypertrophy, we may get some fiber splitting in them. The best that we can tell in humans, the data does not support that, unless maybe if you take steroids, if you take anabolic steroids, you might get some hyperplasia. Okay. The problem with this is that it's hard for us to know, right? Because anybody want to volunteer to let one of my grad students, we're going to cut out your bicep and we're going to dissect it and count every muscle fiber. And then we're going to try to put it back in and let you train. And then we're going to cut it out again and count them again, right, in that way. So it's really tricky, right, in humans for us to be able to try to count what this is. And so what they do basically is they've counted fibers in cadavers of relatively sort of younger people, some that have trained, some that haven't trained, and we try to get a sense of those numbers. And it doesn't seem that the data support all of that. Okay. But this is the percent change in the number of fibers in lower animals. I think these data are from rats. But if we do an overstretch model in rats, we can get more fibers. Exercise, maybe just a little bit, and then this was there's sort of other ways of inducing what we would call compensatory hypertrophy. So if anybody asks, so the question is just in people, we don't think that we split our fibers. So that means you're born with every muscle fiber you're ever going to have. And the way that your muscles get bigger is that we add protein into the existing fibers. We think this may play some role in the genetic limit on how big your muscles can get. Okay. Some of you may have just been born with a higher number of small fibers, which means if all we can do is make each fiber get bigger, right, then you may have a greater potential to hypertrophy than somebody that was born with a smaller number of fibers, but they're already kind of big. Does that make sense, guys? So this is one of the things that we think may play some role in the genetic limit, but it's really, really hard for us to experimentally test. Okay. So this is just from the textbook trying to illustrate what we think happens in humans, right? That you've got 4 fibers, we train them, you still have 4 fibers, but each of those fibers has gotten bigger, okay? It's gotten bigger. And we have 2 things that happen as they get bigger. We're going to increase myofibrillar cross-sectional area, so more actin and myosin, the fiber gets bigger, but we also increase the number of nuclei that we have. Okay. Remember that muscle fibers have many, many nuclei, and as they get bigger, they're going to need more nuclei to supply all the proteins and things for those fibers. And so that's going to happen. You're going to get bigger fibers and more nuclei. If you can't get more nuclei, you're not going to be able to get as big of fibers. Okay. So what are the cellular pathways? Right, was a biology major as an undergrad. I love nothing more than a good cellular signaling pathway. Right, you can memorize this. This thing acts on this thing, and this is a G protein, and we phosphorylate here, and this goes over there, and et cetera, et cetera, et cetera. Okay, so this is an illustration. It was published in 2011. So the illustration itself is roughly 15 years old. We know more even now, but this was in 2011, a kind of cartoon of these are the signals that we know of that play some role in activating protein synthesis in response to exercise. And so there's just a shit ton of things, okay? Just a huge number of things. I'm not going to require you, Ty's not going to give you a worksheet on this. Okay, I'm going to have to memorize all of these things, but I want you to know a couple of important signals, and I've put them here in this red box. Okay, in the red box, especially this one right here called mTOR. Okay, I want you to know mTOR because this seems to be kind of the hub, right, of integrating all of these different signals together and then telling the nucleus to make us more actin and myosin. mTOR stands for the mammalian target of rapamycin. Mammalian target of rapamycin. Rapamycin is an antibiotic. So if I bathe your muscles in rapamycin and have you exercise, they will not hypertrophy. And so that's kind of how we discovered some of these things. All right. mTOR, when it gets activated, most of these pathways that are going to lead to hypertrophy, especially this AKT pathway that's going to come from insulin and insulin-like growth factor and things, signal through mTOR, which signal through something called P70S6 kinase, which then is going to initiate making messenger RNA for actin and myosin. Okay. So when you resistance train, the big takeaway is it activates mTOR. When you take anabolic steroids, especially testosterone, it activates mTOR. Okay? It activates mTOR. And I can mention it here, and I don't know if I have the data in there later, but one of the scientists that was a part of the early discoveries of the usefulness of mTOR now works at, we're not supposed to call it the Health Science Center anymore, whatever in the hell it is, the health campus or something, works at the health campus in Oklahoma City. Her name is Sue Bodine, and she's a brilliant scientist, but was one of the early, she studies muscle hypertrophy and compensatory hypertrophy and all the cellular signals, and was one of the leaders that helped the discovery of mTOR. Okay. So how do we get mTOR to get activated? What is it that's special about exercise that turns on mTOR? What is the signal or signals that lead sort of after you do a bout of exercise that lead to mTOR getting turned on? Okay. Well, one of those signals, one and its importance is not as great as we used to think it was, is an increase after a bout of exercise of what we call anabolic hormones. Okay, testosterone, growth hormone, and something called IGF-1, which stands for insulin-like growth factor 1. We'll talk about this one in a second. And IGF-1 comes in a couple of different variants. One's released from the liver, one's released from the muscles themselves that are going to be really, really important. Okay. We know that certain hormones, if we give them to you above and beyond what you normally have in your body, it will induce muscle hypertrophy. Okay. Or if we give it to you and then you resistance train, it will induce additional muscle hypertrophy. Only been a couple of studies because it's ethically not great for us to give you these things because they cause side effects. But if I give you testosterone in what we call hypogonadal men. So a lot of what we know is we give these hormones in people that don't make them. So hypogonadal men are men that do not make normal physiological levels of testosterone. Okay. So they're low. And these are not sort of men that are of my age or a little bit older that are going through andropause that had testosterone and it has dipped and now we're giving it back. Right. This is not what you see in if anybody watched the golf this weekend or anything, that's all you hear about is right, drugs and things like this. But we get a 15% increase in muscle mass and about a 50% increase in protein synthesis. And they do not work out. You just sit there. We give you testosterone and you get bigger. Okay. Very, very powerfully anabolic. In one of the only a couple of studies in humans that we have, when they gave it to eugonadal men, so men that had normal testosterone, they gave them testosterone and they sat on the couch and they got anywhere between like 10 and 20% bigger muscles. Didn't do anything, just sat there, muscles got bigger. When we combine eugonadal men getting or women getting testosterone with resistance training, we don't know what the limit is, right? We don't know. But you will absolutely get huge. Okay. If you have supra physiological levels of testosterone, and you combine that with resistance training, it very, very powerfully activates mTOR and you get big muscles. And it seems to counteract a lot of the genetic limits and things that are going on. Similarly, growth hormone, if I give you growth hormone, you will get bigger muscles. Growth hormone is not quite as powerfully anabolic from a muscle standpoint as testosterone, but it does tend to lead to the breakdown of fat. So very often people that take steroids will do what they call a stack. They will take both together. They will take the testosterone to get big, and they will take the growth hormone to lose the body fat on those ways. It's also difficult for us to extricate these things. If I give you testosterone, it leads to an increase in production of growth hormone from the hypothalamus. So it's a little bit difficult to tease out what is what. It does seem like that if you take growth hormone and you train, the best data that we have is it's not any better than just simply taking the growth hormone, unlike the testosterone. Okay. Both of these things can be very, very dangerous. Okay, very dangerous. You know people that are taking growth hormone because the classic thing it used to be, and I'm sure there's still a bunch that do, in Major League Baseball, they would take them and their hat size gets bigger. Growth hormone grows bone. So you get sort of your skull and things will change shape. Your head gets bigger. Your feet get bigger. Okay, your feet get bigger. My wife would want me to tell you that while she was pregnant, that her shoe size went up. So we had to buy her all new shoes, which she has thought was just delightful to be able. And so depending upon what it is, it's up like a half or a full size. A very, very common thing, right? Ladies, when you're pregnant, there's growth hormone going around, and your shoe size will get bigger. So if you like your shoe collection now, may want to back things up if you're going to choose to do all of that. So anyway, those things can be really, really dangerous if we take those in that way. Insulin-like growth factor 1 is an interesting one. Okay. So IGF-1 binds to a receptor. It looks to your body like insulin, but it's not insulin. It does not come from the pancreas. It is not released in response to an increase in blood carbohydrate level. So there's the regular variant that comes from the liver. Then your muscles also release the regular variant, and then they release one that only comes from the muscle, which we have termed mechanogrowth factor, or MGF. Okay? Mechanogrowth factor specifically is really, really good at activating satellite cells, those stem cells in skeletal muscle. Okay? You work out a muscle, that muscle releases mechanogrowth factor, which activates the satellite cells that exist in the muscle that was just working out. It helps with repair, it helps with increasing muscle size. It acts very often as what we call an autocrine. Okay? It's released by a tissue and it flips right back around and it binds right back onto those tissues. Whereas testosterone and growth hormone are much more, they're endocrine, that they are released into the blood and they circulate systemically. And as you guys will see in the next thing, the reason that the exercise helps is that when we exercise and we resistance train, we make more receptors for testosterone and growth hormone and IGF-1 in the muscles that we're actively working out, which is why, right, when you work out your arms and you release all of these things, your arms get bigger, but your legs generally don't. It's very specific. We got to upregulate the receptors by working out. Okay. Okay. So what is it about resistance training that leads to the increase in these hormones? Well, there's nothing that's that shocking. If you want to release testosterone or growth hormone, really, you should go run rather than resistance training. You get a much bigger, or at least an equally big, response from running as you will from doing resistance training. But we know there's something about producing force. Running can be very, very high force, okay? Stretching the muscle and the total amount of muscle mass used. So working out bigger muscles, working out total body kinds of things, okay? Stretch and load. So we know that when you generate force and that force gets transmitted across the surface of the muscle fibers through the tendons, through the connective tissue, that stretches and moves around the sarcolemma. It moves around the membrane of those muscle fibers. And that mechanical stress, that movement on the outer membrane, we don't fully understand why, triggers, okay, the release of the hormones and the upregulation and tells that cell to make receptors for those hormones. That disruption of homeostasis, that sort of jarring of the membrane, cell doesn't like. So the response is, let's make more things and let's fix and strengthen up our cell and make it bigger and stronger. Okay, so that's what's going to go there. And then we talked about the upregulation of the receptors. Okay, so you need to load the muscles. You need to stress the muscles. And if possible, right, which most things that we do, how else might you stretch a muscle during a bout of resistance training? Well, you pick the thing up and you set it down. So you do concentric movement and then followed by the eccentric movement. There is some limited data out there that a concentric eccentric movement induces more hypertrophy than a concentric movement alone. You say, well, Dr. Black, what if I just do eccentric? And I'm like, then things get kind of wild and crazy if we just do eccentrics. You get lots of muscle damage, you're sore all the time, and it may diminish some of our returns on all of that. But most people you can't ever train concentrically anyway. Like you need a partner. Like I'm going to pick the curl up and somebody's going to take it from me, and then I'm going to put my arm down and pick it up again. So very practically, most work that we do is concentric eccentric, which helps us get load and some stretch as part of all of those things. Okay. All right. So we talked about this. So I want to be very clear about what the data tell us here, even though I've just spent multiple slides telling you that the hormones are important. because they are. So 20 years ago, when I started doing this, the best science led us to think that the larger the acute change in hormones after a single bout of resistance training, the larger the potential for you to get bigger muscles from that kind of training. Okay. And so because of that, people made certain choices on how they train and what they did. You can especially manipulate rest periods and manipulate how much growth hormone you get. What we know now, 20 years later, and this was a little controversial to start with when it first came out about 10 years ago, but we know we have much more data now, is that the magnitude, okay, the magnitude of increase in anabolic hormones after one bout of exercise plays very, very little to no role in the amount or the size of the muscles that you're going to get afterwards. Okay. The acute increase doesn't matter. What matters is there is an increase. Okay. What matters is that you have these hormones naturally anyway, and you have some ability to increase them. All right, so we've got some things there. We get small increases in IGF-1 and growth hormone afterwards, right? And high levels can support resistance training induced hypertrophy, right? We don't have to have those big kind of things. We have lots of data now where they will do things and blunt the increase, and people still get hypertrophy, or they'll manipulate the training in ways where they don't get as big of an increase. So they're important, but they're important as long as you have them. If you are if you're a woman going through menopause or a male going through andropause, or someone that naturally has lower levels of these hormones, then it can have some effect in that way. But if you've got sort of normal physiological levels, the increase after one bout is not nearly as important. Okay. So I mentioned that mechanogrowth factor, that muscle specific variant of IGF-1, plays a big role in activating satellite cells. And we talked about satellite cells the very first week of class. You have these muscle specific stem cells in every muscle fiber. Okay. And we know, even though they're a stem cell and you think it's a stem cell, it's going to play a role in repairing the fiber and things of that nature, which it does. They are wildly important for inducing hypertrophy. And so because of that, being able to turn them on by getting this proper MGF response is really, really important. And we know that resistance training activates the satellite cells, tells them to divide, and they incorporate themselves into existing myofibrils or myofibers, and they help add nuclei. And that may be one of the big things that they do, is they help us add these additional nuclei, which will then allow the support of those fibers to get bigger and bigger. Okay. Addition of new nucleated fibers is required to support your ability to maximize all of this. So one of the ways that we know that satellite cells are important is through these animal models of studying these things. So we do terrible things to animals in the name of science, and most of it is probably justified, right, in those ways, but it still makes me feel really, really icky. So what you see here, okay, is this is data from, I believe these are mice, and they take these mice and they zap them with radiation, and they irradiate them, and it kills the satellite cells. Okay, I don't really understand how we kill the satellite cells, but not the other ones, but that's what happens. Kill the satellite cells. Then they take the mouse and they cut basically but they cut its gastroc. So it can't use its gastroc, but it can still use its soleus to do what, in the mouse equivalent of plantar flexion. That's called ablation. Okay. It's called synergist ablation. So you would take on them, they've got two muscles that basically work together to do plantar flexion. You cut one of them, so you put a lot more stress and load on the one that's existing. And so what they do then is they do the ablation, and you get about a 16% increase in the size of the soleus in these animals over a period of time. Okay, we irradiate them, no ablation, but we just do irradiation and their muscles get smaller, just resting muscle, they lose muscle because they have no satellite cells. Then we do the irradiation and the ablation. So we zap the satellite cells, we cut the muscle, so we should get this much of an increase, and instead they get smaller. And so from doing that, we know then that the satellite cells play this really, really important role in looking at everything. Okay. So IGF-1, MGF help us activate the satellite cells. There's probably also something about mTOR that helps us activating the satellite cells. These are some of the earliest data on if we block mTOR, we can block hypertrophy. This is from the woman who now works at the Health Science Center that I said, from Sue Bodine. And so this is basically, right, what we would expect at 7 days and 14 days. Okay, they've got a compensatory hypertrophy model. They're cutting the muscle again. Okay, they're getting this like 25% increase in muscle size in 7 days. Mice grow really fast. Their time course is all out of whack with us. Okay, but they then cut and then they give the mouse rapamycin and they blunt this increase dramatically. So if we can't activate mTOR, we don't get nearly as much hypertrophy. Here we are out here at 14 days, right? We're getting even more hypertrophy. And with the rapamycin mice, we're still blunted, okay? So satellite cells matter. mTOR really, really matters. And they then also looked at, is the mTOR activation specific to one fiber type or not? This is the control. Here's the compensatory hypertrophy in those fibers. And here are those fibers where the rapamycin has been applied. And so what you can see is that basically activating mTOR in both slow twitch and fast twitch fibers leads to hypertrophy. Blocking the mTOR, and you're going to get a relatively similar blocking of hypertrophy in both. Okay, let's see here. We know that in general, most of you can make your type 1 fibers and your type 2 fibers hypertrophy. There's a little bit of an issue that because we use our type 1s a lot more often, right? Especially those ones that are recruited at low force, you have basically already hypertrophied those fibers as much as you're probably going to. And so in a lot of instances, the fibers in your body that have the highest capacity or the highest ability to get bigger are the ones you don't use very often. And so lots of times those seem to be type 2X fibers or the last of the type 2As, but the signaling pathway at least works in all of them. Okay, it works in all of them. So how does exercise stimulate mTOR? Although Veer, you had a question, but let's do the question.
Yeah, I was gonna ask, why does type 1 fibers' hypertrophy not look like anywhere close to as similar as like type 2's? Like,
What do you mean?
like marathon runners or like ultra marathon runners look like really skinny compared to like sprinters.
Sure. You're assuming that ultra marathon runners have a lot of type 1 fibers and that sprinters have a lot of type 2, right? So, but a lot of it, if I told you guys, when you go run a marathon, you get a giant testosterone and growth hormone release. And yet, you don't get bigger muscles. So there's something specific, and part of it's about metabolic turnover and the way the receptors and things are going to be upregulated in the midst of all of that. And so I would say that the marathon runners look the way that they look in a lot of instances because of all of the training and the diet and the sort of high metabolic turnover. It's hard to be anabolic when you run that much, even when you eat that much in that particular way. And a lot of them don't resistance train very much. Although we do have good evidence that from a neural perspective, if they will do power cleans and things like that, that it actually improves their movement economy and it makes them better distance runners. But some of that is self-selection. Some of it is also, it's the nature of the metabolic response during everything. It's not that their type 1 fibers don't have a capacity to hypertrophy. It's that the nature of their training doesn't sort of elicit that hypertrophy. Because you could take a marathon runner and we could have them stop running. We could just, make them a gym bro, and we can make them hypertrophy. So most of them. Okay, that's a really good question, Bear. It's a really, really important kind of thing. Okay, how does exercise stimulate mTOR? The honest answer is we don't fucking know. I'm sorry, we don't. Like we've been searching for this for for years, and we have some ideas. We think that when you eat protein, that helps to stimulate mTOR eventually. When you get the hormone response, the testosterone and growth hormone and IGF-1 eventually will elicit an mTOR response. Okay. But we also know that there are these sort of yet undiscovered, what we imagine are called mechanotransducers. There are things in the membranes of your muscle fibers that sense that load, that sense the force, that sense the disruption in the fiber from the stretching and the high force, that signal in some way, shape, or form down here to mTOR, and that it very likely is the combination of all of these things together that work to activate mTOR. Okay, it's the combination of all of those things that activate mTOR. Okay. So we talked about anabolic steroids, but we talked about how they're going to work with mTOR, they're going to work with satellite cells, but there's sort of an anti-growth factor. It's called myostatin. Okay. All of you have, well, I say all, most of you, probably everybody, has some amount of myostatin in your body. And the myostatin acts like a governor in that it limits how big your muscles can get. Okay. And so what you see here, this is called a Belgian blue bull. And this is a bull or a cow, right? Well, I guess it can't be a cow. It's actually technically a bull, but it's a bull, does not make myostatin. This bull has never lifted a weight in its life. And yet, he is swole. He has no myostatin. None. You want to see something crazy or scary or whatever, just on your things, Google myostatin deficient dog. Google myostatin deficient rat. Okay. These animals all exist. They just naturally, some of them have very, very little myostatin. We have people that have very low or no myostatin. This is basically a myostatin deficient child. And what I can tell you, for you guys that don't know, maybe you've not been around little kids, 7-month-olds, you should not be able to see their vagus lateralis. They should look like the Michelin tire man. Okay. Just there, they're all squishy, and everything. It's great. But we have people. There are people, noted Olympians that basically have no myostatin. Okay. So one of the things that we think places a genetic limit on how big your muscles can get is your naturally occurring amount of myostatin. Some of you have less myostatin, therefore you may have a greater capacity to hypertrophy your muscles. Some of the people that basically can get no hypertrophy, no matter what they do, may have very, very high levels of myostatin. So now, if we block myostatin artificially, we get better muscle growth. I'm not deep in the discord with people that take steroids and all of those things, but I guarantee you that there are sort of off the market drug companies and things that make compounds that block myostatin. You probably go to Steel and talk to somebody in the locker room, and they can hook you up with something that might block myostatin. Question on all of those things is, how safe is that? Right, your muscles get too big, it outstrips the ability of your ligaments and tendons to do things and other things. But this is another piece of something that works to govern how big your muscles are going to be. Okay. I don't like looking at the myostatin deficient dogs. They make me scared. It's like something out of a nightmare. Okay. So we'll finish off just a little bit talking about some of the genetic influence, as I have said, right? One of the big things that determines who can get big muscles, which most people can't. Like, you can get them bigger, but most of you are not going to get and look like The Rock or whatever. The Rock doesn't look like The Rock. Just Google 1998 The Rock when he was first wrestling versus now, and it's all steroids. Okay, so about 80% of the differences in muscle mass between individuals is because of genetic variation. By contrast, about 40% of the difference in VO2 max is genetic. So about 80% of muscle is genetic. We know what the genes are. Okay. Those of you that have done 23andMe, swab the inside of your cheek and it will tell you these things. We can identify if you have the certain alleles that might predispose you one way or the other to be able to have bigger muscles. As far as we know, there's about 47 genes that we know of right now. Many of them are linked directly, almost all of them are linked directly to mTOR. Okay. Large difference. We have these high responders and low responders. A lot of it is about genetically your ability to activate mTOR. That balance of fiber size, of myostatin, of receptor density, and the ability in satellite cells and everything to kind of work through the mTOR pathway. Okay? That's what we know. Lots and lots and lots of sort of interperson difference is driven very, very simply by genetics. Okay. There's been lots of research done, a lot of it out of the medical center at the University of Alabama at Birmingham, where they group people, they do these big studies and they group people as sort of non or low responders, as people that are kind of a moderate responder. You're, basically like less than 5% muscle growth in that, like 10 to 20% growth as a moderate, and the people that are over that, they're high responders. And they've looked at sort of differences in gene expression of those key genes across them. And we see, right, that there are some very, very clear kind of genetic predisposition types of things for all of that. Okay. Okay. Our very, very last slide. Doing okay. This is what happens when my throat hurts and I don't feel great. There's no stories today. I'll tell you a story about this one. And we've spent a week talking about when I train and I squat and I do whatever, my muscles get big. Okay. They get big. What we don't talk about, that will come for all of you in your life at some point, very likely, is that when you don't resistance train, when you don't move around, your muscles get smaller. We call that atrophy. Okay. Atrophy. So there's been a lot of research, initially a lot of it driven by NASA, about what happens during spaceflight on disuse of skeletal muscle or unloading of skeletal muscle. If you guys want to make a lot of money, I think you can go to Houston right now and get enrolled in NASA is constantly running these bed rest studies. It puts you in a hospital bed, you get a little bit of like 8-degree head down tilt because that's better for your blood pressure. And you're on bed rest for 8 weeks, 12 weeks, 16 weeks, 6 months. Oh yeah. And then sometimes while you're doing the bed rest, they give you different drugs. They'll have you do different exercise things while you're laying down. One of our ways to induce atrophy or to mimic spaceflight is to put you on bed rest. Okay. We know that when you don't use a muscle, fiber size decreases both fast and slow. We do, if it's super, super, super unused, you will get conversion from slow to fast. You really have to have like a spinal cord injury or something where you're completely immobile. Force production goes down to a greater extent than mass, because you lose not only the mass, but you also lose those neural adaptations. Lots of this is driven by a decrease in protein synthesis. You become catabolic from doing all of this because you lose your anabolic things. When I was a grad student, the guy that I worked for, before he came to Georgia, had worked for NASA. And they studied all of this in the 80s. They studied, like, we're getting ready to send people to the International Space Station. All of the stuff that we do now was sort of in the infancy stages. And so he studied muscle atrophy. And they developed a way where you would walk around, give people a set of crutches, you give them a giant platform to put on one shoe. So like a 10-inch platform on your left foot, on your right foot, you had to be able to drive. And crutches, and you would crutch around. We had people crutch around for a month where their left leg just would swing around. They did not bear any weight on their left leg for a month. Okay. We paid people $1,000 to do this. Every one of them said, no fucking way I would ever do it again. You need more, right? We recruited a bunch of people weirdly from a this is less of a thing now from a call center. Where they just sat around all day making like random, sort of pestering phone calls anyway. But in a month, we had them do that. Their calf lost about 18% of its muscle mass. Their quad lost about 15% of its muscle mass. You go to space, right? Within like 2 days, we are already seeing measurable decreases in size. That's why if you ever see stuff from the people on the International Space Station, they spend like 8 hours a day exercising. And also it's hilarious to try to figure out how do you run on a treadmill when there's no gravity? How do you lift weights when the weights don't weigh anything? Right? They use rubber bands, they strap them down onto the treadmill. The treadmill like doesn't have a motor. You use your own your own when you push on it, it will make it run. But we know when we don't use muscle, it atrophies. Even if you have hormones, it still atrophies. There's something about if you lose your hormones, it will atrophy some, even if you can't hypertrophy as much, even if you're resistance training. But it's very much about the loading and the resistance training and stuff. When that stimulus is taken away, we get atrophy. Okay, Wednesday we start metabolism. So here we go. All right, very good. Thanks, guys.
Don't forget, there's a worksheet due tonight. Tomorrow the answer will be posted, so make sure you go by and review your answers for the worksheet, guys. Thank you.
No, that's.
Yes. Yeah,